Hello and welcome to Chapter 18, Biodiversity, Classification, and Conservation. Mockingbirds are found throughout the Americas. There are 4 species of mockingbird in the Galapagos Islands in the Pacific, of which the Floriana mockingbird is the rarest. It became extinct on the island of Floriana in the second half of the 19th century, mostly because people colonized the island and introduced species like rats, which destroyed its nests. People also cut down the prickly pear cactus, which is the bird's source of food and a favorite nesting site. 2 small populations remain on nearby tiny islands. Conservation programs have had some success at increasing the numbers of this species, and there are plans to reintroduce the species to Floriana. One of the rarest birds in the world, it is worth saving from extinction, not least because when Darwin visited Floriana in 1835, he noticed that the mockingbirds on different islands within the Galapagos were not all alike. The rest is history. Diversity can be thought of as a catalog of all different species. So when a new species is discovered, it is given a scientific name using the binomial system that was developed by Linnaeus in the 18th century. Each species is given 2 names. The first is its genus and the second species. The 2 names together are indicated species, or its scientific name. A species is a group of organisms with similar morphology and physiology, which can breed together to produce fertile offspring and are reproductively isolated from other species. So for animals, most plants and fungi, the term species is used to mean a group of organisms that appear similar to each other and that can successfully breed together. Around 250,000 flowering plant species and thousands of other plants, such as ferns and mosses, are known. Add to this the fungi, bacteria, and other single-celled organisms, and we are looking at a very large number of species. Even though amateur and professional biologists have been cataloging species for hundreds of years, there are still places on Earth that have not been explored fully, such as the oceans and tropical forests. One tree in a tropical rainforest can contain as many as 1,200 species of beetles. Almost every time biologists do a thorough count of beetles in a small area of rainforest, they find many previously unknown species. Also, scientists don't agree on the species that have been described and often subdivide them or group them together. Estimates of the total number of species range from 100 million, range up to 100 million species. So species do not live in isolation. They share their living space with others to form communities. These communities of organisms interact with each other and with their environment. A forest ecosystem includes not only the community of organisms, but also the soil, the dead leaves that form leaf litter, water in the rain and in the streams, the air, rocks, all the physical and chemical factors which influence organisms. No ecosystem is entirely self-sustained. Organisms in one ecosystem interact with those from another. Many birds, for example, migrate from one ecosystem to another, often over very great distances. An ecosystem is a relatively self-contained, interacting community of organisms and the environment in which they live and with which they interact. So a small pond is an ecosystem, as the water provides a very different environment from the soil and air around it. The organisms in the pond are therefore very different from those of the surrounding ecosystems. At the other end of the scale is the open ocean, which is a huge ecosystem. Some ecosystems are very complex, like tropical rainforests and coral reefs, and others are very simple, like a sandy desert. So a habitat describes the place where a species lives within an ecosystem. The niche occupied by a species is more than a physical description of its place. A niche is the role of an organism within the ecosystem. It is how the organism fits into that ecosystem. An organism's niche describes where it is, how it obtains energy, how it interacts with both its physical environment and with other species. Organisms require resources from their surroundings, and they have special adaptations for obtaining them. In many ecosystems, there are similar niches that may be occupied by the same species, or, more likely, by different ones. In Australia, the niche for large herbivorous animals is filled by kangaroos. So on the savanna of Africa, there are zebra, antelope, wildebeest, elephants, and others that fill those large herbivore niches. They coexist because there are differences in such things as the type of vegetation they eat. It is almost impossible to provide a complete description of the niche of any organism because there are so many ways in which one organism interacts with other components of the ecosystem. A niche is the role of an organism in an ecosystem. Biodiversity can be defined as the degree of variation of life forms in an ecosystem. This is usually taken to include diversity at 3 levels: the variation in ecosystems or habitats, the number of different species in the ecosystem and their relative abundance, and the genetic variation within each species. So some areas of the world have very high biodiversity. Examples would include the Congo Basin in Africa, Southeast Asia, the Caribbean and Central America, Amazonia, and Southwest Australia. These areas have many endemic species, that is, species that are only found in these areas and nowhere else. Species diversity. So the number of species in a community is known as species richness. Species diversity takes species richness into account, but also includes a measure of the evenness of abundance of the different species. So the more species there are, and the more evenly the number of organisms are distributed among the different species, the greater the species diversity. So coral reefs have a very high biodiversity. Such an ecosystem offers many different ecological niches, which are exploited by many different species. Species diversity is considered important because ecosystems with high species diversity tend to be more stable than those with limited diversity. Okay, they are able to resist changes. Some ecosystems are dominated by 1 or 2 species, and other species may be rare. So this is the case in the natural pine forests in Florida and the temperate forests in Canada, which are dominated by a few tree species. The tropics are important centers for biodiversity, possibly because living conditions are not too extreme. There is light of high intensity all year round, and birds and mammals do not need to expend energy keeping warm. So for example, there are about 1,500 species of birds in Central America, but only 300 in the Northwest Territories of Canada. Genetic diversity is the diversity of alleles within the genes in the genome of a single species. All the individuals of a species have the same genes, but they do not all have the same alleles of those genes. Genetic diversity within a species can be assessed by finding out what proportion of genes have different alleles and how many alleles there are per gene. The genetic diversity that exists, so you can see here, so here is like one type of kiwi. Here's the one that we're probably the most familiar with, but look at all of the genetic diversity. So the genetic diversity that exists between varieties of cultivated plants and domesticated animals is obvious because we can see the differences, right? So like here, looking at these potatoes. Similar genetic diversity, although not always obvious, exists in natural populations. The genetic differences between populations of the same species exist because populations may be adapted slightly differently in different parts of their range. There is also genetic diversity within each population. So this diversity is important in providing populations with the ability to adapt to changes in biotic and abiotic factors such as competition with other species, evading new predators, resisting new strains of disease and changes in temperature, salinity, humidity, and rainfall. So here, when we're looking at diversity within a species, we're looking at the importance of variation for natural selection to work. Collecting organisms and making species lists is a way to assess species diversity. So imagine you're in an ecosystem like this one here. The most obvious species are the large plants and maybe some of the larger animals, particularly bird species. The first task when assessing species diversity is to identify and catalog the types of organisms and build a species list. So biologists use identification keys to name the organisms that they find. There are different forms of keys. Some have drawings or photographs with identifications. Others ask a series of questions. The most common of these is a dichotomous key. In your own fieldwork, you may use one of these to identify some plant species. Identification requires good skills of observation. So at first, it's a good idea to do a timed search throughout the area you're studying to see how many species you can collect and then identify. So here's an example. Here's an identification guide to freshwater macroinvertebrates. You can see here that there's the jointed legs and then no jointed legs, and then the different types of identifications that you can make within each of those categories. Okay, if you can't identify a particular species, you take photographs of them, you name them as species A or B. Some animals will be hard to find and collect, especially small ones. So a pooter is a simple piece of apparatus that is used to collect them. It's a clear jar that is easy to see through. It has a lid, and through the lid are two tubes, one for the collector to suck through and the other to take the species into the examination chamber. The tube you should suck through should have a little piece of gauze across the tube so you can't accidentally inhale whatever you're collecting. Breathing air into the mouth sucks up small animals into a plastic container. They can then be removed and studied and identified using a hand lens and then returned to their habitat after. Here's an example there. Okay, sampling. So to find out which species are present in an ecosystem and the size of the population to each of them, the ideal method would be to find, identify, and count every single organism that lives there. Sometimes you can do this if the area is really small or the species is really large, but it's only rarely possible to do that. So instead, we take samples from the area we are interested in and use these to make an estimate of the total numbers in the area. Sampling can be random or systematic. If an area looks reasonably uniform or there is no clear pattern to the way species are distributed, then it's best to use random sampling. A quadrat is a square frame that marks off an area of ground or water where you can identify different species that are there or take a measurement of their abundance. So you need to decide on a suitable size for your quadrat and how many samples you'll take. Samples must be taken randomly to avoid bias. So for example, you might choose to take all of your samples from the place with fewest species simply because it's easier to do. This would not be representative of the whole area that you were surveying. So the usual way to ensure that a sample is random is to mark out an area with measuring tapes and then use a random number generator like an app or one on your mobile phone. The random number gives you the coordinates of the sampling points in relation to the two tapes you have used to mark out the area. You can use your results in two different ways: to calculate species frequency and species density. Species frequency is a measure of the chance of a particular species being found within any one quadrant. You simply record whether the species was present in each quadrant that you analyze. For example, if you place your quadrat 50 times and found daisy plants in 22 of your samples, then the species frequency for daisies is the number of times a plant species is present in a given number of quadrants of a particular size or at a given number of sample points. Okay, so here is your example. So it'd be 22 divided by 50 times 100 is 44%, right? So species density is a measure of how many individuals there are per unit area. For example, the number of individuals that you have counted is divided by the total area of all your quadrants. It is not always possible to count individual plants and animals because of the way that they grow or their size. Many animals and plants grow over surfaces, forming a covering that's almost impossible to count individuals. So in this case, you can estimate the percentage cover of the species within your quadrat. To help with this, you can use a 100 centimeter by 100 centimeter quadrat with wires running across it at 10 centimeter intervals in each direction, dividing the quadrat into 100 smaller squares. You then decide approximately what percentage of the area is occupied by each species. These percentages may not add up to 100%. So for example, there might be bare ground in the quadrant, so the numbers will come to less than 100%. Or there may be plants overlying one another, in which it may add up to more than 100%. An alternative to estimating percentage cover of each species is to use an abundance scale, such as the Braun-Blanquet scale for number and plant cover. A method of describing an area of vegetation devised by Braun-Blanquet in 1927. So it's used to survey large areas quickly. Two scales are used. One consists of a plus sign and a series of numbers from 1 to 5 denoting both the numbers of species and the proportion of area covered, ranging from plus, sparse and covering a small area, to 5, covering more than 75% of the area. The second scale indicates how the species are grouped and ranges from 1 to 5. So quadrats are obviously no use for finding or counting mobile animals, so different methods have to be used for these. So small mammals like mice and voles can be caught in traps that are filled with hay for bedding. Insects and other invertebrates like spiders can be captured by sweep netting. Also, pond nets are used for sampling aquatic organisms. The techniques for this vary according to the size of the body of water and whether it's still or moving. Single birds can be counted quite easily, although it does become difficult in dense forests. And flocks of birds are much more difficult to count, although it can be done by counting a group of 10 birds and estimating how many such groups there are. Okay, mark and release. So a good method of estimating the population size of mobile organisms, if you use it carefully, is the mark, release, recapture technique. So first, as many individuals as possible are caught. Each individual is marked in a way that will not affect the future chance of survival, right? So like an ear tag or something if it's a bigger animal. The marked individuals are counted, returned to their habitat, and left to mix randomly with the rest of the population. When enough time has elapsed for the mixing to take place, another large sample is captured. The number of marked and unmarked individuals is counted. The proportion of marked to unmarked individuals is then used to calculate an estimate of the total number in the population. So for example, if you find that 1/10 of the second sample was marked, then you presume that you originally caught 1/10 of the population in your first sample. Your best estimate is therefore that the number in the population is 10 times the number you caught and marked in your first sample. So here is a worked example, the mark, release, recapture technique. So the number of caught in the first sample was 247, and the number caught in the second was 259. The number in the second sample that had been marked was only 16. So you do 247 times 259 divided by 16, right? So then here is the estimated number in the population. Okay, Simpson's index of diversity. So this is a measure of diversity. It can be calculated by using this formula here, okay, where D is the total number of organisms of a particular species, and N is the total number of organisms of all species. Values of D range from 0 to 1. So a value near 0 represents low species diversity. A value near 1 represents high species diversity. One advantage of this method is that you do not need to identify all or even any of the organisms present to the level of species. You can, for example, just decide to call the species of an enemy that has short tentacles an enemy A, and the species that has few long tentacles an enemy B. And so long as you recognize that they are different, you do not need to find their scientific names. Okay, but beware, some individuals in the same species have different phenotypic forms. Okay, so the higher the number we get for D, the greater diversity. You can probably see that the diversity depends on the number of the different species there are, and also the abundance of each of those species. So a community with 10 species, but where only one is present in large numbers and the other 9 are rare, is less diverse than one with the same number of species where all the different species have similar abundance. Comparisons using this diversity index should be on a like-for-like basis so that the community should be similar and the organisms should be also similar. So for example, it should not be used to compare the diversity of fish in a lake with the diversity of moths in a forest. Okay, systematic sampling. So systematic sampling is when samples are taken at fixed intervals, usually along a line. And this normally involves doing transects, where a sampling line is set up across an entire area where there are clear environmental gradients. So for example, you might use a transect to show the changes of plant species as you move from grassland into woodland, or to investigate the effect on species composition of a pollutant radiating out from one source. A transect is a path along which one counts and records occurrences of the species of study. Okay, so this requires an observer to move along that fixed path and to count occurrences along and at the same time, obtain the distance of the object from the path. So this results in an estimate of the area covered and an estimate of the way in which detectability increases from probability 0, which is far from the path, towards 1, which is near the path. A line, so then, yep, you can see here. So here's the transect, and then it's counting the organisms that are found at regular points along the line. So belt transects. Okay, this is a technique in which by placing a quadrat at regular intervals along the line and recording the abundance of each species within the quadrat. So in this method, the transect line is laid out across the area to be surveyed, and then a quadrat is placed on the first mark point on the line. Quadrats are sampled all the way down the transect line at each mark point of the line or at some other predetermined interval, usually randomly. It is important that the same person do the estimations of cover in each quadrat because the estimation is likely to vary from person to person. The abundance of organisms within quadrats placed at regular points along the line is noted. A belt transect will supply more data than a line transect. So it will give data on abundance of individual species at different points along the line, as well as on their range. Okay, so looking at those two sampling techniques that we covered, we looked at random sampling as well as systematic sampling. Okay, so if you're doing systematic sampling, belt transect is a good way to go. So at this point, you can draw some kite diagrams to display data on species abundance. And the kite chart is a perfect diagram to graphically enlighten the way that abundance of various species change at different places along the same line. Okay, so they're used to see trends and statistics in a visual way. They show the change of percentage over distance. The central line for each diagram has a value of 0. The kite is then drawn symmetrically, both above and below the line to represent your data. So for example, you can use kite diagrams to compare the distribution of plant species along a coastline. Okay, so here you see seaweeds and mollusks. Okay, so under seaweeds is kelp, serrated rack, bladder rack, and spiral rack. Here's distance from the shore in meters. Okay, so all the way from 0 to 200 meters. And then you can see in the kite diagram, you're able to see kelp only is in this 0 to 20 meters from the coastline, where you can see the serrated rack is all the way from the coastline to 120, and so on. Okay, so this helps you to get a really good visual data of species abundance according to the line, the transect line. Okay, so, and then you want to use a correlation. So this is a statistical measure that indicates the extent to which two or more variables fluctuate together. A positive correlation indicates the extent to which those variables increase or decrease together in parallel. A negative correlation indicates the extent to which one variable increases as the other decreases. So a positive relationship exists when both variables increase and decrease at the same time, and a negative relationship exists when one variable increases and the other decreases, or vice versa. A scatter plot is then used to explain the relationship between dependent and independent variables. So here we go. So the strongest correlation you can have is when all the points lie on a straight line. But, you don't always get that. So let's look at, here's a negative linear correlation. So as x increases, right, as x increases, y decreases. Okay, negative linear correlation. A positive linear correlation is where x increases, y increases. Okay, and then no correlation is just no line. Okay, the points do not collect around any linear formation. So again, the strongest correlation you can have is when all the points lie in a straight line. Okay, that there is a linear correlation, and this would be a correlation coefficient of 1. So if as variable A increases, so does B, the relationship is a positive correlation. If A increases and variable B decreases, then it's a negative correlation. A correlation coefficient of 0 means no correlation at all. So you can calculate a correlation coefficient to determine whether there is a linear relationship and also to find out the strength of that relationship. So Spearman's correlation coefficient measures the strength and direction of association between two data sets. So here is our equation. D is the difference between each pair of ranked measurements. Okay, we know sigma is the sum of, and n is the number of samples. So how and why to use Spearman's rank? So if you have done scatter graphs, Spearman's rank offers you the opportunity to use a statistical test to get a value that can help you determine the strength of the relationship. So you used you used Pearson's correlation coefficient. You have a graph. You see that there is some kind of relationship, but you want to determine the strength of the relationship between those two data sets, because you're not always going to get this line where all of the data are right on the line, right? There's going to be a spread of data. And so sometimes it might look like there's a linear relationship, but you're not quite sure. And so it's really useful to use Spearman's rank to see the strength of that relationship. Okay, so the Pearson's correlation coefficient is a statistical measure of strength of a linear relationship between paired data. The Pearson correlation coefficient, R, can take a range of values from +1 to -1. A value of 0 indicates no association. A value greater than 0 indicates positive association. A value less than 0 indicates a negative association. Okay, pretty straightforward. So the correlation coefficient can only be used when you can see there might be a linear correlation, and when you have collected quantitative data as measurements, like it has to be length, height, depth, light, intensity, whatever, or counts, so number of plants in a quadrat. Okay, it's a technique for investigating the relationship between the quantitative continuous variables. Like, remember from chapter 17, continuous variables being things like height and weight. Okay, not discontinuous variables. So Pearson's correlation coefficient is a measure of the strength of association between these two variables. So looking at, is there a relationship between height and weight, right? So it gives information about the magnitude of the association or correlation, as well as the direction of that relationship. And the first step is to check whether the relationship between the two continuous variables appears to be linear by drawing your scatter graph. The correlation coefficient should not be calculated if the relationship is not linear. Okay, so if you just see a random scattering. The nearer the scatter of points is to a straight line, the higher the strength of the association. So before using the test, you must also be satisfied that the data for the variables you are investigating show a normal distribution, right? That they're continuous variables. So as trees go older, they tend to get cracks in their bark. A student measured the width of the cracks on many pine trees in a plantation and found that they varied considerably. The data she collected showed a normal distribution. She noticed that the larger and presumably older trees tended to have wider cracks in their bark than the smaller ones. She wanted to see if there was a correlation between the size of the trees and the size of the cracks. She chose to measure the circumference of each tree as a measure of their overall size. Now, this is important, okay, because the width of the cracks as well as the circumference of the tree is continuous, meaning that it can be on a whole range of data points, right? So she collected continuous data for each of the two variables, the tree circumference and the crack width. She investigated this by selecting 12 trees at random, measuring the circumference of each trunk and the widths of 3 cracks on the bark at head height. Okay, so here's the data. So the number of trees, 12, circumference of trees, and the mean width of each crack. Okay, so there is your equation. Okay, so in Pearson's linear correlation, it shows a linear relationship between two sets of data. So sometimes you want to see if there is a relationship between two variables. And so we're looking at the number of species P and species Q in 10 different quadrats. Are the two species related to each other? Okay, so these are two different types of data sets. So the first thing we do, we plot a graph. So looking at this graph, we can see that the data looks as though they might lie approximately on a straight line. It looks as though there might be a linear correlation between them. And to find out whether it's so, we use the Pearson's linear correlation test. Again, this can only be used if the data are interval data and normally distributed. And we can see because they do not appear to change position in any one direction and there are no obvious outliers. Okay, here's our equation. Okay, take a second and look at each of these values. Okay, here's our data. So we calculate x times y for each set of values. We calculate the means for each set of figures, x and y. Okay. We calculate n times the sum of x and y, so that here that's n equals 10, okay, and so forth. We add up all the values. Now we calculate the standard deviation, and then substitute numbers into the formula to calculate for r. Okay, pause this, take a really good look at this. So a value of +1 means total positive correlation between your two sets of figures. A value of -1 means total negative correlation. A value of 0 means there is no correlation. So here we have a value of 0.81. That lies quite close to 1. So we can say that there is a positive linear correlation between the numbers of species P and the numbers of species Q. Okay, so then Spearman's rank correlation is used to find out if there is a correlation between two sets of variables when they are not normally distributed. So as with Pearson's linear correlation test, the first thing to do is plot your data on the scatter graph and see if it looks like there's a correlation. And note for that this test, the correlation need not be a straight line. It doesn't need to be linear. Okay, so let's say you've counted the numbers of species R and species S in 10 quadrats. And here's your number, and here's your scatter plot. Now rank each set of data. So for example, for the number of species R, quadrat 4 has the largest number, so that's ranked as number 1. Go through and order them in that way. Okay. So once you have ranked both sets of results, you need to calculate the differences in rank, D, by subtracting the rank of species S from the rank of species R. Then you square each of those values and add them together. To find the sum of D squared, which in this case equals 12. Okay, so pause this and look again. Look at how they're ranked here. Okay. So the number of species R, quadrat 4 has the largest number, and so that's ranked as number 1. Okay, so you're ranking species R and S in this way. Okay, then you calculate the differences by subtracting the rank of species S from the rank of species R. Then you add them together. Okay, so the formula for calculating Spearman's rank correlation coefficient is here. Okay, this number that we have here, this number is the correlation coefficient. The closer the value is to 1, the more likely it is that there is a genuine correlation. Just like with our other calculation, our value is very close to 1, so it certainly looks as though there is a strong correlation. Okay. So, when to use them? Spearman's rank is for ordinal, non-nominal data. Values are observations that can be ranked, counted, and ordered. Pearson's is for normal data. Okay, values are observations that can be assigned a number, but data cannot be ranked. Okay, so Spearman's for non-normal, Pearson's for normal. Normal meaning its distribution. Okay, examples: how does the population size of grasshoppers vary in relation to when pesticles were last applied, as fewer insects found, where pesticide application is recent? Okay, so again, you're picking a statistical test to find if there's a correlation between data sets. One test you use for discontinuously distributed data, meaning just like counts of organisms, okay? And one you're using for continuous data or normal data, okay? And that's Pearson's linear correlation. Okay, moving on. Classification. Classification is a systematic grouping of living things based on characteristics, hierarchical or phylogenetic relationships. Okay. Taxonomy is the science of defining groups of biological organisms on the basis of shared characteristics and giving names to those groups. We find it difficult to memorize or absorb information about thousands of different unrelated objects. So by grouping them into different categories, it's easier to understand them and to remember their key features. So taxonomy is the study and practice of classification, which involves placing organisms in a series of taxonomic units or taxa. In biological classification, these taxa form a hierarchy. Each kind of organism is assigned to its own species. Similar species are grouped into a genus. Similar genera are grouped into a family, families into an order, orders into a class, class into a phylum, and phyla into a kingdom. The domain is at the top of this hierarchical system. So, how African bush elephants and hibiscus plants are classified. So let's look at these. So for African bush elephants, you're in the domain Eukarya. So is hibiscus. Okay, they're eukaryotic. They're eukaryotic organisms. They have more than one cell, right? They're multicelled organisms. Okay, but then they diverge right there. In the kingdom, elephants are in the animal kingdom, Animalia, and the hibiscus are in the plants. And you can see as it goes down, okay, all the way down to the species level. And you can see at the species level, the species is named after its genus and then species. Okay. So kingdom is the largest and most inclusive grouping, right? Plant, animal, fungi. Phylum are organisms constructed on a similar plan. So that's, like having a backbone, right? Chordata or not, okay? Class is a grouping of orders within a phylum, so mammals or not. Order, a group of apparently related families, right? Like primates, okay? Family, a group of, I already said that. No, a group of apparently related genre, okay? So like hominids within that, or like New World monkeys or Old World monkeys. Genus, a group of similar and closely related species, okay? So for our case, that's Homo. And then species is a group of organisms, so Homo sapiens. Okay, 3 domains. So biologists used to divide organisms into 2 large groupings based on their cell structure, and this would be prokaryotic and eukaryotic, and they have significantly different cellular structures. In the 70s, prokaryotes were discovered living in extreme environments, like hot springs where the temperatures exceeded 100 degrees Celsius. And these extremophiles, as they are known, were not like other bacteria, okay, other prokaryotes. So studies revealed that the genes coding for the RNA that make up their ribosomes were more like those of eukaryotes. And they were found to share features with both typical bacteria and eukaryotes. So at this time, studies of molecular biology assumed a much greater significance. Okay, this meant that a new taxon, the domain, had to be introduced to reflect the differences between the extremophiles and the typical bacteria. So the domain is the taxon at the top of the hierarchy. So the prokaryotes are divided between the domains Bacteria and Archaea, and all the eukaryotes are placed into the domain Eukarya. Okay. So here we go. So where it used to be eukaryotes and prokaryotes, which we still understand, we can still understand a lot of organisms based on this, but it's really not complete enough. And so now we have 3 domains: Bacteria, Eukarya, and Archaea. So many Archaea live in extreme environments like hot springs and volcanic vents. Some of them produce methane, cannot survive where there is oxygen, and many have unusual enzymes. And since they were discovered in these extreme environments, they have been found in many less extreme environments as well, like they form an important part of the plankton in the oceans. And in several ways, the Archaea appear to have more in common with Eukarya than bacteria. It's thought that bacteria and Archaea separated from each other early in the evolution of life, and then Archaea and Eukarya probably diverged later. Bacteria are prokaryotic, as their cells have no nucleus. They are small organisms that vary in size between that of the largest virus and the smallest single-celled eukaryote. The characteristic features of bacteria are they have cells with no nucleus. Their DNA exists as a circular chromosome and does not have histone proteins. The smaller circular molecules of DNA called plasmids are often present. No membrane-bound organelles. The ribosomes are 70S. They're smaller. The cell wall is always present. The cells divide by binary fission, and they usually exist as single cells or sometimes small groups of cells. The filamentous Cyanobacterium nostoc. Okay, so these species fix carbon dioxide in photosynthesis. It also fixes nitrogen by converting N2 into organic forms of nitrogen in the wider, lighter green cells in its filaments, which is obviously super important for all other forms of life above them. Okay, archaeans are also prokaryotic, and their cells have no nucleus, so their range of size is similar to that of bacteria, and most of them inhabit extreme environments. Okay, they have cells with no membrane-bound organelles. Their DNA also exists as a circular chromosome without histones. They have smaller circular molecules of DNA called plasmids. They also have the 70S ribosomes, but they have features that are similar to those in eukaryotic ribosomes, not as close to the bacterial ribosomes. Their cell wall is always present. Their cells divide by binary fission, not mitosis. And they also usually exist as single cells or small groups. Okay, the metabolism of archaeans, though, is similar to that of bacteria, but the way in which transcription occurs has much more in common with us, with eukaryotes. So a scanning electron micrograph of the Archaean molecule here. So although they look like bacteria, archaeans have differences in their metabolism and their genetics. The flagella seen here is different. Okay, it's only found in near boiling water, and if the temperature falls below 70 degrees Celsius, it dies. It'll freeze and die. It respires anaerobically using sulfur instead of oxygen, which is super interesting. So all the organisms classified into the domain Eukarya have cells with membrane-bound nucleus and organelles. Their characteristic features are they have cells with a nucleus and membrane-bound organelles. The DNA in the nucleus is arranged as linear chromosomes with histone proteins. Ribosomes are 80S in the cytosol. Chloroplast and mitochondrial DNA is circular. There's a greater diversity of forms. They use cell division by mitosis, and there are many different ways of reproducing, asexually and sexually. So here is a protocystian covered in many cilia, which it uses for movement and for feeding. And although it's unicellular, it has considerable specialization of regions within its body. So the Protoctista kingdom is made up of a diverse range of eukaryotic organisms, which includes those that are often called protozoans and algae, like seaweeds. Any eukaryote that is not a fungus, plant, or animal is classified as a protocyst. The characteristic features, so eukaryotic, mostly single-celled, or exist as groups of similar cells. Some have animal-like cells with no cell wall, and are sometimes known as protozoa. Others have plant-like cells with cellulose cell walls and chloroplasts and are sometimes known as algae. So here's an example. So the body is composed of thousands of cells within the flagella. They work together in a coordinated way, but there is little specialization of cells. The cells at one pole detect light, so then the colony swims towards it. Cells at the other poles are specialized for reproduction. Inside there are new colonies that are just about to be released. Many organisms in this kingdom may actually be more closely related to organisms in other kingdoms than they are to each other. So for example, there are strong arguments for classifying algae as plants. Okay, fungi. So fungi have some similarities with plants, but none of them are able to photosynthesize. They are all heterotrophic, obtaining energy and carbon from dead and decaying matter or by feeding as parasites on living organisms. There is a vast range in size. A specimen here, this is of the honey fungus. It grows in a forest in Wisconsin and spreads massively. Not only is it possibly the largest organism in the world, but it may also be the oldest. Okay, characteristic features of fungi are eukaryotic, do not have chlorophyll, do not photosynthesize. They use heterotrophic nutrition. They reproduce by means of spores. They have a very simple body form. Okay, they have cells, or their cells have cell walls that are made of chitin or other substances, not cellulose, and they never have cilia or flagella. Here's an example here. So the bread mold fungus and a detail of it. So here it's another example, and if you look really closely, that's when you start to see it coming off of the surface of the bread. Fascinating. Okay, so plants are all multicellular photosynthetic organisms. They have complex bodies that are often highly branched, both above and below ground. So the characteristic features of plants, they are multicellular eukaryotes with cells that are differentiated to form tissues and organs. There are a few types of specialized cells. Some cells have chloroplasts and photosynthesize. Cells have large, often permanent vacuoles for support. They are autotrophic nutrition. Their cell walls are always present and are made of cellulose. And they occasionally have flagella. As an example, some of the oldest trees on Earth here. Okay, the kingdom Animalia. These are multicellular organisms that are all heterotrophic with many ways of obtaining food. There is a great diversity of forms within the animal kingdom. The nervous system is unique to the animal kingdom. So the characteristics are multicellular eukaryotes, many specialized cells, cells that are differentiated to form tissues and organs, and cells do not have chloroplasts and cannot photosynthesize. Although some, such as coral polyps, have photosynthetic protocysts living within their tissues, cell vacuoles are small and temporary. They are heterotrophic nutrition. Their cells do not have cell walls. Communication is through the nervous system. And cells sometimes have cilia or flagella. Okay, viruses. Viruses are microorganisms whose structure is only visible with electron microscopes. Viruses are acellular. They do not have acellular structure like bacteria and fungi. You will have noticed that some viruses are not in the classification system we have discussed so far, and that's because viruses have none of the features that we traditionally use for classification. Okay, so there is an argument that they should not be considered to be living organisms at all, but they do have particles made of proteins and nucleic acids that are found in other cellular organisms. When they are free in the environment, they are infectious, but they don't have a metabolism. When they infect cells, they make use of biochemical machinery of their host cell to copy their nucleic acids to make their proteins, often leading to the destruction of the host cell. The energy for these processes is provided by respiration from the host cell. Here's a model showing the structure of part of a tobacco mosaic virus, the TMV virus. The blue strand is the single strand of RNA, and the rest is composed of just proteins. So in a survey of plant pathologists in 2011, TMV was voted the most important plant virus. So viruses are classified into 4 groups using the type and structure of their nucleic acid as the criteria for grouping them. So you can see nucleic acid, DNA, RNA, number of strands, example, host organism, and disease. So it's interesting the type of nucleic acid that they use. They can use DNA or RNA. They have 1 to 2 strands. Okay? They have a range of different organisms and a range of different diseases. All right, that is the end. Thank you for listening.